In our last lesson, we considered the structures and properties of amino acids that were uncharged at neutral pH. Let's look at those that are charged and see how they compare. There are two amino acids that we classify as acidic, glutamic acid and aspartic acid. These carry end carboxyl groups in their side chains, the distinction being the length of the aliphatic portion of their side chains. These are acidic groups that readily release protons at neutral pH to form the carboxylate or conjugate base form. This is why we often refer to them as aspartate and glutamate. Note that it is the low pKa values of the side chains that makes these acidic amino acids. In contrast to these, we have three amino acids that we refer to as having basic side chains. Pictured on the left is lysine, which as you can see carries a long aliphatic hydrocarbon chain. However, it carries an end amine group at the epsilon position. This amino group has a high pKa value that readily accepts a proton at neutral pH, thereby becoming ionized. In the case of arginine on the right, it also has an aliphatic chain, but the terminal functional group is a guanidino group. It has the highest pKa value of any of the amino acids and is therefore always charged at physiological pH. The side chain of histidine is an imidazole ring that can be protonated. Its pKa value is actually slightly less than neutral, yet it can be protonated close to a pH of 7 and is therefore considered a basic amino acid. Let's consider for a moment the acid and base properties of every amino acid. Each has an alpha carboxyl group whose pKa is on the average of 2.2, though individual values vary due to the nature and influence of the side chain. If we compare this pKa to that of acetic acid, whose pKa is 4.76, the alpha carboxyl group is a much stronger acid. That is, it more readily releases its proton. Why is this true? It's because of the electron withdrawing effect of the alpha amine. Remember, they are very close to each other, with only the central carbon atom between them. By comparing the alpha amine group with a secondary alkyl ammonium ion, we see that the alpha amine is a weaker base, and that has to do with the influence of the nearby carboxyl group and its electron donating effect. The important lesson here is that pKa values for functional groups are influenced by the local environment. If we compare the guanidine group of arginine's side chain, we find it a much stronger base than an aliphatic amine. That is to say, the pKa value is much larger. This is because of resonance stabilization after accepting a proton. A unique feature of the amino acid histidine is that its side chain imidazole is a heterocyclic aromatic amine. Thus far, we've considered the structures and properties of the 20 common amino acids. It is perhaps important to point out that there are some other amino acids that are uncommon but derive from the common amino acids. These are often produced through modifications that occur after synthesis, referred to as post-translational modifications. Examples include hydroxylysine and hydroxyproline, pictured here. Thyroxine is a modification of the amino acid tyrosine and is tissue-specific, found only in the thyroid gland. We'll see some examples of proteins containing these modified amino acids later. For now, just realize that there are amino acids other than those we consider the 20 common amino acids.